Today we have Dr. Josie Bryant to speak to us, and she's a group leader at the Parasites and Microbes program at the Wellcome Sanger Institute. So she uses genomics to investigate microbes in people's lungs, how this respiratory microbiome evolves and adapts over time, and how this affects human health and drives disease. So in today's talk, she will talk about her research on a little known but important bacterium, Mycobacterium abscessus. So many bacterial pathogens live both in people and the environment around us. So it's often unclear where people have caught that infection, but genomics has revolutionized our ability to track the movements and evolution of bacterial pathogens. And this can be used to transform our understanding of Mycobacterium abscessus, which causes life-threatening lung infections. So without further ado, please put our hands together for Dr. Josie Bryant.
So I'm a microbiologist and also a molecular biologist, and throughout my career I've mainly been using genomics to understand how bacteria evolve. So I'm going to try and tell you today by giving you an example from one pathogen about how we can understand how bacteria evolve on lots of different evolutionary scales, from individual cells to people to hospitals to continental transmission. So an important thing that you really need to appreciate is that bacteria are constantly mutating and adapting to the environment around them. So you're probably all aware of the COVID pandemic and the fact that you have variants of COVID that have spread around the world and outcompeted other variants. And this has all happened via mutation. And this is exactly the same for bacterial pathogens as well. And so one really nice example of this is experimental evolution, which we do quite a lot for bacteria. And one example of this is Richard Lenski's long-term evolution experiment, which he's been doing with E. coli in Michigan State University, where he's been growing E. coli since 1988, so the same strain. And he's been growing it over time, over thousands and thousands of generations, and seeing how E. coli has been adapting and competing and evolving into new strains and going through these processes called selective sweeps, and even patterns of hypermutation where you suddenly have rapid accumulation of lots of mutations. And these experiments have been really good for telling us about how evolution actually happens at a really, really fine scale. But all these processes happen in the real world as well. And the thing I really like about bacteria is that they happen at all sorts of different scales, and you can study them at all these different scales. So you can look at single bacteria, so you can actually do single cell sequencing of bacteria now to look at how they mutate and how they interact with each other. You can look how they mutate and diversify at the scale of a whole organ, and so how they diversify over the lungs, for example, during an infection. You can look how they mutate and evolve at the scale of the whole body, and how they adapt to different lungs in our bodies and the very different environments found there. And then you can look at outbreaks, so how they transmit between people, and how the bacteria may be evolving to become more or less transmissible, and try and understand why that might be. And then on from that, you can then try and understand how certain strains of bacteria have adapted to become so pathogenic that they're spreading all across the world. And the example I'm going to give you today is going to touch on all these different evolutionary scales. So the other thing that I really like about bacteria is that most bacterial pathogens are actually opportunistic pathogens, and that's because they primarily live in the environment. So they mainly live in soil and water, and it's only when a susceptible person comes into contact with them that the bacteria is able to then cause an infection and live within that person. And so there's loads of really interesting processes going on here, in that you're trying to understand how a bacteria can survive in both the the environment and the soil and water and how it can survive in people as well. They're very different environments. And there's also lots of research going on understanding how some bacteria have managed to make the jump from being environmental to being purely disease-causing pathogens, and what the mutational processes were that led to that. And one example of a bacteria that's made that jump is Mycobacterium tuberculosis, which only causes disease and is only ever found within people and only passes by person-to-person transmission. So Mycobacterium tuberculosis belongs to the genus Mycobacterium, but today I'm going to be telling you about Mycobacterium abscessus, which is actually, so here's a tree that I built of all the housekeeping genes across the genus. And so it's the opposite end of the genus, and unlike TB, it does live in the environment, and it's thought to be in aquatic environments, and it's actually found to be really adept at infecting and living within amoebae. And the way that it's managed to survive in amoebae is actually thought to explain why Mycobacterium abscessus is actually pretty good at living in human macrophages as well, which are a really important part of our immune system, and they're responsible for chomping up any foreign material, including pathogens. And so Mycobacterium abscessus causes disease in people because they can survive within human macrophages, which are usually intended to destroy the pathogen, but Mycobacterium abscessus actually lives quite happily within them. And so Mycobacterium abscessus causes very nasty respiratory infections and also soft tissue infections as well. That's the name of abscessus, because it causes abscesses. So I'm going to be telling you about what we've learned about Mycobacterium abscessus today, particularly in the context of cystic fibrosis, because people with cystic fibrosis are very susceptible to these infections. And then I'm going to tell you about what we've learned about how it transmits, how it evolves over the course of an infection, and then finally take all these bits of information together and show you our evolutionary model we propose for Mycobacterium abscessus. So cystic fibrosis is an inherited lifelong disorder which causes defective mucociliary clearance in the lung, which makes people with cystic fibrosis very susceptible to respiratory infections. And they go into this cycle of infection which causes inflammation and lung damage, which then makes them even more susceptible to infection. And it's actually with these infections which cause death in cystic fibrosis patients. And if you have CF and you're infected with Mycobacterium abscessus, you're actually likely to have a more severe decline in lung function than if you were infected with some of the other common pathogens for cystic fibrosis. And these infections are becoming worrying because rates of infection are increasing worldwide, and it's very, very difficult to treat. So many of the drugs that we would treat with TB, Mycobacterium abscessus is already resistant to it. So there's very, very little treatment options, and Mycobacterium abscessus can actually evolve over the course of an infection to become resistant to these drugs. And so rather than relying on sort of these population level statistics, I sometimes find it's useful to focus in on individual patients, because I think this paints a clearer picture for what's actually, what these infections are actually like. So this is a case history of one patient. It was a lady born in 1986 who had certain mutations that caused her cystic fibrosis disease. And then around the age of 10, she was found to be infected with a mycobacterium, and it wasn't until a few years later that they positively ID'd it as Mycobacterium abscessus, because the microbiological identification of these organisms can sometimes be quite tricky. So she received antibiotics which should be effective against Mycobacterium abscessus, and so she started off having sporadic courses, and these intensified over time. And despite receiving these antibiotics, she declined and had a very poor lung function, and the infection wasn't clearing, so she was referred for a double lung transplantation in 2011. And so these were her X-ray demonstrate images before and after the transplant, which I don't really understand, but I'm told the fuzziness in the one on the left is indicative of lots of lung damage and inflammation and infection, and it looks quite clear after a transplant. And she then goes through a period of time where she is doing relatively well and she feels a lot better. And by a year later, she starts declining again, and this is a PET CT scan, and these hot patches are bits of inflammation and infection, where it's thought that the Mycobacterium abscessus was re-establishing itself, and they found that she was again positive for Mycobacterium abscessus. And then it even got to the stage where it had spread out of her lungs and had formed these lesions on her skin, these little abscesses. And her symptoms became intolerable, and she accepted end-of-life care, and she died. So this is not an unusual case. This happens in a lot of these cases, and so I thought this illustrates this quite well. But also, we've actually learned a lot about Mycobacterium abscessus just by studying this one patient. So I'm going to return to her later in the talk. How did she acquire an infection, and how generally people pick up Mycobacterium abscessus infections? So I mentioned that Mycobacterium abscessus lives in aquatic environments, and previously it was thought that people picked up infections by being exposed to water somehow, and then, but then after that there wasn't any onward transmission between people. It was only environment to person, and that there wasn't any onward spread between people. But we were approached by Pathurst Hospital during my PhD, and they had noticed an increase in cases in their cystic fibrosis unit, and so they asked us if we would sequence their Mycobacterium abscessus genomes, and at this point there was only one Mycobacterium abscessus genome available in the world, so we thought it might be interesting. And what we actually found was that many of the isolates, when we looked at their genomes, they clustered into two branches on the tree, and that within these clusters they were very, very highly related, and given the mutation rates and the sort of phylogenetic structure, it indicated that these were transmission clusters. And I've been doing work on TB genomics at the time, and it looks remarkably like TB when we knew that transmission had been happening between TB patients. So we thought this was evidence of transmission, and this had happened for 11 out of the 30 patients were infected with Mycobacterium abscessus in the clinic. The interesting thing is that there wasn't any evidence that any of these patients had any direct contact with each other. So we think instead it was indirect person-to-person transmission, and that perhaps they were transmitting it to each other through surfaces, so these are called fomites, inanimate objects that you might cough on and then someone else might pick up the infection from that, or infectious aerosols, because many of these patients have physiotherapy at hospitals, so they generate aerosols, and maybe the other person enters the room after that and picks up the infection somehow. Because although they hadn't had any direct contact with each other, we actually had evidence that many of them had been in the hospital on the same day, so maybe this was a mechanism of how they spread to each other. So this was quite a major finding, but we still didn't really understand how people were spreading Mycobacterium abscessus to each other, and it's actually very difficult to work out. Can't go back and work out people's exact movements around the hospital, what they touched and what they didn't touch. So, but there was one case where we managed to work out what was going on, and this was a single occupancy room at Papworth Hospital, where in 2013, patient 1, who was Mycobacterium abscessus positive, had stayed in there from April till September. So this is a single occupancy room, so it's just got a bed and a sink, and I think it might have a shower as well. They were then discharged, and the room was cleaned like it normally would, and then patient 2 came and stayed in October, then they left, and then patient 3 stayed from November to December, and then were discharged, and then patient 4 stayed in late December. So patient 2, 3 and 4 by the end of December was still deemed Mycobacterium abscessus negative. But then in January they found that patient 4 was positive, and patient 2 then became positive in February, and patient 3 became positive in April. And they then did a little infection control investigation, and they swabbed the whole room, and the only thing that they found was positive was the sink trap, which is the bit, you know what I mean, the drain of the sink. And we did genome sequencing and found that they all had identical genomes. So this suggests that maybe patient 1 had coughed in the sink, and then somehow this had spread to patient 2, 3 and 4. But what was curious is that we assumed that the room had been cleaned many, many times over this time period. But we actually swabbed it again in June, and again we found the same strain again, even though it had been intensively cleaned between this period. And it wasn't until we changed to bleach, because Mycobacterium abscessus is actually resistant to many cleaning products. We changed it to a much stronger bleach that it ended up being negative Mycobacterium abscessus. So in this case, we think that people have transmitted this bacteria to each other via the sink. So you can imagine that they coughed in the sink, it grew a biofilm in the sink trap, and then when someone else turns on the tap, it creates an aerosol that they then breathe in. And that's why some microbiologists said you shouldn't leave your toothbrush near your toilet, because when you flush your toilet, it will create aerosols that might have in your toothbrush. Our work directly led to a change in infection control policy in the US, UK and Australia. It led to changes with how patients are separate, and make sure they're kept separate within the hospital environment if they're positive for Mycobacterium abscessus, and it also directly impacted on the design of the new Papworth Hospital, which isn't in Papworth anymore, but it's at Addenbrooke's, and they had much more up-to-date ventilation and more air changes, which should hopefully reduce aerosolized transmission of pathogens. And it's actually been incredibly useful for the COVID pandemic, as many patients were treated with. And so we then wanted to know, was this just a rare case? Was it just that there was lots of transmission at Papworth? So we then went on to collect samples from all the cystic fibrosis treatment centres across the UK. We got samples from the US, across Europe and also Australia, and we found that this wasn't a rare case, and that we saw the same sort of phylogenetic patterns indicative of transmission across most of the centres. But we found a bit more than this. We found that when we put all the isolates that we had collected from across all these different locations together, and we built a phylogenetic tree, we identified the 3 subspecies that were already known to exist for Mycobacterium abscessus. But we noticed very sort of clear patterns on the tree where you have some isolates which are very far apart from each other, like in Mycobacterium abscessus villeti. But then you have some parts of the tree where you have these very tightly clustered clones, and these isolates, when you zoomed in, weren't close enough for you to say, oh, it's an outbreak, but they were much more closely related than the rest of the tree. And so we called these dominant circulating clones, and these sort of dominant circulating clones are known to exist for other bacteria as well, and they're sort of hypothesized to be clones that are more successful in humans, so they're ones that may be moving towards the more human disease niche and away from the environmental niche. That's the hypothesis anyway. And we found that these dominant circulating clones were found across all the 3 continents that we had samples from. And when we did experiments in the lab with both just cells and also mice, we found that they were more virulent. And when we looked at the clinical outcomes of patients, the clones that are becoming more adapted to the disease than the rest of Mycobacterium abscessus. The other thing that was really interesting is that we were able, when we had more isolates a few years later, we were able to do Bayesian-based gating analysis, which is where you take the trees and you use that to estimate how old these clones are. And we actually found that remarkably many of these clones had a very similar age around, that we think they had a rapid expansion around the 1960s. And when you look at the history of treatment of people with cystic fibrosis, you actually see a rapid increase in life expectancy around the 1960s, and it's actually corresponded to the first cystic fibrosis specialist treatment centres being opened in the UK and the US. So these treatment centres had a remarkable positive effect on people with cystic fibrosis, and that they were getting more specialised treatment, and thus they lived longer. But also these treatment centres provided opportunities where these patients could mix and actually pick up spread infections to one another. So it might actually provide the opportunity for these clones to evolve. And so after that, we then wanted to know whether we could look at the genomes of these clones and see what makes them different from the others. So usually in bacteria, when you do this, if you look at particular successful clones that seem to have changed niche, you're hoping that if you look across them, they all share the same plasmid, or they all acquire the same gene. It ended up being more complicated than that, but what we did identify, so this is a pan-genome analysis where we basically reconstructed the genetic content of all the Mycobacterium abscessus cases, and these kind of knots are where you have bits in the genome where there's lots of swapping of genes. And through this, we identified that in the dominant circulating clones there was an enrichment of genes involved in global changes in global gene expression, and also modifications of DNA. And we actually, one that caught our eye was this methyltransferase, which hadn't been described before, which was found in one of the more worrying dominant circulating clones, which was responsible for many of the outbreaks, including the one at Papworth. And so we decided to test whether the acquisition of this methyltransferase had made this dominant circulating clone more virulent. And so we knocked it out, and we did PacBio sequencing, which is a way of identifying methylation patterns, and we found that across the whole genome it did have a different methylation pattern. And then when we also did RNA-seq and found different patterns of gene expression, but we also infected mice and also macrophages in the lab, and found that acquisition of this gene led to higher rates of survival within macrophages. So we think that this gene may be contributing towards the higher levels of virulence in this dominant circulating clone. So we haven't managed to explain all of it, but we sort of have beginning to piece together understanding what makes these clones different. Well, one of my main interests now is understanding within host evolution, and that's because many of these patients, as I said, they won't respond to treatment, so they'll be infected for decades, or basically until they die. So they'll be infected for years and decades, and so there is a very long opportunity for the Mycobacterium abscessus, once it's infected someone, to then evolve further within that person. So I wanted to understand this process. And to do this, we usually, when you sequence bacterial genomes, you pick individual colonies, and each colony represents a single bacterium in theory. Instead of doing this, we took plate sweeps where we cultured the Mycobacterium abscessus, and then we scraped off as much of diversity as possible. And this means that we're not just looking for just consensus mutations, we're looking for low frequency or minority variants, which is something before this had only really been done in cancer and on viruses. So I was adapting a lot of methods used in other fields for this. And what I found was that I could detect quite a few minority variants, and when I plotted how they changed over time within individual patients, I saw that many of them followed the same trajectories over time. So skipping many, many months for me to work this out, I eventually worked out that I could use statistical clustering to cluster the variants into different clones circulating within the patient. And then by looking at the relative allele frequencies over time, you can actually infer how they're related to each other. And I also use a bit of graph theory called transitive reduction. But essentially what you do end up with is the subclones that are within a patient and how they vary over time, and then how they're related to each other. And you end up with a phylogenetic tree. And so this is an example from one patient where each of the blobs represents a different subclone, and the branch lengths represent the number of mutations between them. And then you can then use that tree to reconstruct the evolutionary history within this patient. So this is just a single patient over time, and you can see from all the different colours and how they change really rapidly that it's very dynamic. So there's a lot of diversity, and there's a lot of rapid changes over time, which is quite surprising for a bacteria that is supposedly not supposed to have a very high mutation rate. And we found out loads of really interesting things from that, including what happens when they're given a new drug, and working out specific pathways to drug resistance, and we could see evidence of hypermutation. But one of the most striking findings was parallel evolution, and that's, when I say parallel evolution, I mean within a single patient there were independent lineages picking up the same kinds of mutations. And so in this example, when you have the red dots, it's that in 4 different lineages, 4 different non-synonymous, so amino acids changing mutations happened 4 different times within the same person. And they're not happening on top of each other, so it's not an accumulation of variants, it's they're happening independently. And my hypothesis is that this is happening because the lung the anatomy of the lung is very structured, it's very compartmentalised, there's lots of branching points, and so you end up with lots of little pockets of infection. And assuming they're under the same selective pressures, it means the same kinds of mutations are happening multiple times independently in different parts of the lung. And so I observed this pattern for basically every single patient that I looked at. So I looked at about 20 different patients, and I kept seeing parallel evolution, which is a very strong sign that there is very strong selective pressure happening. And this is just a diagram across 18 patients, and it's arranged concentric circles around the genome of Mycobacterium abscessus, and the size of the blobs indicates the number of mutations that I've observed. And so we found the mutations in the same genes happening over and over again in lots of different patients. And we know from tuberculosis that many of these genes that are also found in TB are known to be important for virulence in TB and also drug resistance. So all kind of fit with a camera. We've done a keyhole surgery basically, and they all acquired Mycobacterium abscessus infections, and we still don't understand how this is spread across. Investigating it at the time, and so we had all these genome sequences, and we found, so we looked to see if these same mutations were also happening in these surgery-related infections, and we didn't find any of these mutations a single time, which, given the number of isolates we had and the amount of diversity, that's not just a chance or a sort of statistical issue. So we think that this is strong evidence that the mutations we were seeing here in respiratory infections are due to a selective pressure that's only happening in the lung and it's not happening in other parts of the body. And we also found that many of these genes were related functionally, they're all part of the same functional just using String, which is a database of functional networks of genes. And so I identified the 5 genes with the most number of mutations across the most number of patients, and then I generated CRISPR knockdowns of these genes, and then infected macrophages, and found that all of the knockdowns had increased survival in human macrophages. So we think this is evidence that these mutations are causing the Mycobacterium abscessus to survive even better in macrophages, and therefore are more able to cause disease and be more virulent. So this was really interesting. Once I discovered these mutations, I then wanted to go to the rest of the data set and see, do these mutations accumulate more widely, and are they then spread? So could they, for example, mutations in these genes, could they explain why these dominant circulating clones that I mentioned earlier, why they've become more successful? I actually found the opposite, which was that Mycobacterium abscessus, when it picked up these mutations, actually seemed to be less likely to then go on and transmit to other people. And so there's sort of a transmission fitness cost associated with its adaptation. So by surviving better in macrophages and causing more disease, it actually means that they don't seem to transmit as well, don't spread on to other people as well. And this was something that's being observed for other chronic viral infections, so particularly HIV, where it's been found that there's rapid diversification within a host, but it's actually the ancestral strain that hasn't gone under all the diversification that actually spreads onto other people. And it's been hypothesized it's the same thing, that by rapidly adapting to one person, that it means that you then can't spread on to other people as well. But we've gone one step further than HIV, and we actually managed to experimentally show that this, we think, is something to do with fomite-mediated transmission, it's a surface-mediated transmission, because we found that strains that picked up these mutations were less able to survive on a surface, but then they had exactly the same growth phenotype in sort of liquid culture, but if we just laid them out on a dish on the lab bench and left them, they seem to survive less well compared to strains without all these mutations. So we think that this adaptation has led to a specific fitness cost in terms of fomite-mediated transmission, which we think is a particularly important way that that strains are able to be spread between each other, for example, like in the sink scenario. So we've now generated a lot of information on Mycobacterium abscessus, and so we started sort of putting all these bits of information together. And so we think that people acquire Mycobacterium abscessus from the environment, and maybe some strains, such as the dominant circulating clones, are more able to be acquired than others because they've had horizontal gene transfer of transcriptional regulators and DNA modifiers like the methylase. And then once they get into people, they are then able to undergo rounds of more adaptation, and there's a selection for increased survival in human macrophages. And we think that this process is accelerated by parallel evolution, which allows more different types of mutations to occur in different parts of the lung. And then we saw a sort of surprising finding, which was that within host, adaptive variants were less able to transmit. So even though we've shown now that there is transmission between people, we think that the sort of the adaptive processes don't always promote this process. And so there are a couple of unanswered, sort of frustrating things to do with this. First of all, it bothered me that we saw parallel evolution, and I hypothesized it was due to the anatomy of the lung, but we hadn't actually shown that. So I wanted to show that. And secondly, this sort of mechanism, we do see lots of transmission of Mycobacterium abscessus between people. So I wanted to understand how that was happening despite this adaptation having a fitness cost. These sort of conflicted with each other. And actually, by studying the lady that I mentioned at the beginning, we actually have begin to understand a bit more about this. So as I mentioned, this lady, patient CG, had an infection, she was treated for antibiotics, it failed, so she had a lung transplant. Over this, most of the period where she had her infection, we actually had samples over this whole time, so we could look at the Mycobacterium abscessus genomes and see how they were evolving over this time. And what we found was that there was lots of different subclones present, but at the beginning of her infection there was a whole array of different subclones that were all completely drug sensitive, so these antibiotics were able to be effective. But then it didn't take long for these fully drug resistant clones to then evolve, which were actually fully resistant to both aminoglycosides and macrolides. So I find it particularly depressing that in the last years of her life, her Mycobacterium abscessus were actually fully resistant, and yet she was given really intensive therapy with very, very strong antibiotics, which have all sorts of side effects. And really, they weren't doing anything because her infection was already completely resistant. Because she had a lung transplant, we actually had access to her explanted lung tissue. So these were the old lungs that they took out at the time of transplant. And we had the tissue bank had stored sections from all over her lungs, and we extracted DNA from them. And usually what we would do is grow up Mycobacterium abscessus and then genome sequence it, but because it was fixed tissue that had been kept in formaldehyde, everything was dead, so instead I had to design a base to do a different way of sequencing so that we could pull down specifically Mycobacterium abscessus DNA. And so we did that, and we identified in most of the biopsies from different parts of the lung, we could identify Mycobacterium abscessus, and we identified 3 different clones. 2 of them were unsurprising because they were found in her sputum samples around the time that she had the transplant. And what was also striking is that most of the biopsies only contained 1 subclone, so this was evidence of compartmentalisation of the infection. But what was surprising is that in biopsy 12 we actually identified 2 subclones, and one of them, the pink one, we had only seen in her sputum quite a few years before, and so it was a bit confusing why it was there. But we think this is evidence that more ancestral, less adaptive clones can actually survive in the deep parts of our lung, and that there can be pockets of unadapted clones, and this might explain why transmission of sort of more ancestral clones is able to occur despite all this adaptation happening. So just by looking at this one lady, we've shown that there's compartmentalisation because most of the time we only see 1 subclone per section, and also we think that this provides a possible explanation for how transmission is able to occur in the face of this adaptation, and that you have some kind of you have diversity maintained over the lung tissue, so the clones that are more able to transmit are still present. And so we now sort of have a more complete picture, so we think that M. abscessus transmits between people, but there's still this sort of evolutionary bottleneck which prevents sort of further pathogenic evolution. But you could hypothesize that given the right scenario, the right population size perhaps, that maybe it would be able to overcome this barrier and evolve into a full pathogen. And I've shown here a picture of Mycobacterium tuberculosis, which, as I said at the beginning, is a full is entirely a human pathogen, and it's really interesting that it's been shown for Mycobacterium tuberculosis that it's also in its evolutionary history acquired transcriptional regulators and DNA modifiers by horizontal gene transfer, and also accumulated mutations in the exact same genes that I found have evidence of parallel evolution. And it's thought that this process happened around 6,000 years ago. And that maybe one day it could happen for Mycobacterium abscessus. So I'll finish there. So most of this work I carried out during my Wellcome Trust Fellowship at the LMB